Moving to our last lecture of our school, we have Jean-Martin Beaulieu from University of Toronto, and he will be speaking something about matryoshka in space.
Hey, you just landed the crew, cinema crew, so matryoshka in space, why not?
Привет, Мартинский.
So you can share the screen.
Okay, I'm gonna start. And then
Привет, Эрик, привет. Doesn't hear me.
I hear you, привет.
Ну вот, давно бы сказал.
Okay, so yeah, we can see your presentation.
You
Yeah,
still see it? Great. So now can I see is my question. Okay. So, yeah, fantastic. So integrating scale in psychopharmacology, hence the pathway, circuit, and matryoshka in space. And the reason for the matryoshka is that you have something within something, within something, within something, etc. And so what we're interested in is mental illnesses, which are a major public health issue. Actually, they do represent, um, okay, I have to get rid of this. Goodbye. Good, perfect. They do represent about 17 to 20% of global public health burden. And the problem with that is that, well, when we talk about mental health issue, what we have is disease that are defined by symptoms, not by causes. Actually, DSM is one of the go-to book for diagnosis, and this book was created to provide American insurance company with disease category that they could put in their organigram and calculate reimbursement. That's the origin of DSM. So, which means that the symptom defined disease also define treatment. And if we want an idea of how bad is that, imagine somebody is coughing, then of course the symptom define the treatment, therefore the treatment is cough syrup. That's clear, that's simple, that's go-to. Of course we know that coughing is not cured by cough syrup, it's just lessening the symptoms, and that if you're coughing persistently, you could have several things that could be respiratory, that could be gastrointestinal, that could be others, and then tests will be done, x-rays will be done. These days, viral detection test will be used, and then you could have COVID or you could have lung cancer. And honestly, that's not going to be the same approach to treat you. So that's a bit of a problem with mental illnesses because we don't know what we're addressing. So where do we stand on that? Well, unfortunately, we're a little bit stuck. So the field is going from burst to stall. The last burst was about 2014 when a large-scale genetic study allowed to identify common genetic human variants that were associated with mental illnesses, in this case schizophrenia. And these things are all over the board. So these are some example of gene from that publication from 2014. And to cut the long story short, but some genes are involved in glutamate neurotransmission, calcium signaling, neurodevelopment, synaptic plasticity, ion channel, microRNAs, etc. And all these genes are not exactly highly penetrant. All these variants are not highly penetrant, meaning that carrying any combination of these is not very good as a diagnosis tool. It increases the risk, statistically valid, but it's not very usable. But it instructs us a bit about, for example, schizophrenia. So yes, schizophrenia is probably affecting glutamate neurotransmission. Now somebody like Raoul will tell you, yeah, we know that since the 1980s. Hey. So how do we go from that to drug targets is remaining a big question because several of these genes are not, or gene product are not obviously druggable or could have effects all over the place, and we don't know when, and we don't know how, and we don't know what side effect we will cause if we go after these targets. So outcomes, we are having therapies because we are having therapies. These are just examples of molecule prescribed for schizophrenia or to be prescribed for schizophrenia. And outcomes, we have all these, but the main reason is that we've been lucky. Some of these compounds were given to people and by serendipity gave effect, or given to animals and by serendipity gave effect. And these effects were interpreted as probably it's going to work in patient. And that's where we stand. So essentially, instead of having the rational way of doing research, which is to go from basic research, understand the disease, do translational research to develop therapies, and then do clinical trials to prove these therapies are working. For the case of antipsychotics and pretty much everything that is prescribed in psychiatry, uh, we started with the clinical trials, not necessarily very well managed or very ethical clinical trials. It explains that most of the drug principles were discovered in the 1950s, and after that it stopped. Then, through a process of reverse translation, essentially figuring out how these drugs may work, we got some ideas about the disease, which may or may not fit with what the genetic is telling us. So, for example, in the case of schizophrenia, most antipsychotics are in antagonists of D2 dopamine receptors. That led to the dopamine hypothesis of schizophrenia, which is that there's too much dopamine in the brain of people with schizophrenia, which is probably partially true, as shown by brain imaging studies, but it's not at all fitting with what the genetic is telling us. I mean, we don't have an obvious hyperproduction. I mean, people with schizophrenia don't overexpress tyrosine hydroxylase and they don't produce more dopamine, basically. Changes in dopamine levels is probably part of the disease process, but is it the root cause? We don't know. So, targets for most of these drugs are belonging to a non very original class of drug targets, which are G protein coupled receptors. And G protein coupled receptors represent the vast majority of drug targets. As you can see here, 70M1 is so about 30 or more percent of drugs on the market are targeting G protein coupled receptors. So, not extraordinary original and not telling us that much about what the diseases are doing. So in the lab, we were interested in trying to understand one of the last drugs that nobody is understanding, but that is working, which is lithium. Originally, the active ingredient in 7 Up, the lithiated lithium soda. Lithium was pulled out from 7 Up when people realized that it was actually causing kidney failure and that it may not be a good idea to have lithium in the food chain. However, the drug is lithium as a drug is prescribed, is working very well. And one of the main suspected target, because we do not have a well-defined target for lithium, is a kinase named glycogen synthase kinase 3. So the effect of lithium on glycogen synthase kinase 3 was discovered in 1996 by a guy named Peter Klein. And essentially, what's happening is that lithium is a direct inhibitor of GSK3, and GSK3 is regulated by several other signaling pathways. For example, the kinase AKT is phosphorylating GSK3 and inhibits it. So, and that may happen downstream of BDNF receptors. So, this is all interesting. So, what did we do to address the question of lithium? But a long, long time ago, when I was postdoc and had almost no gray hairs, we were interested in the interaction, the effect of lithium on the dopamine neurotransmission. And what we've shown is that in that knockout mice, so mice that are not that different, but probably a little less clever than the that knockout rats you saw this afternoon, uh, we add an effect of lithium on the locomotor activity induced by increased dopamine neurotransmission in these mice. And it turned out that this led us to the discovery of a new mechanism of cell signaling for the D2 dopamine receptor, the target of the antipsychotics, which involves beta-arrestin and PP2A, which in turn inactivate AKT, which prevents the inactivation of AKT by GSK3, and GSK3 is now activated by the D2 dopamine receptor, meaning that lithium would inhibit GSK3, inhibit this cascade, and that antipsychotics would inhibit D2, which would also inhibit this cascade. So that's an interesting cascade, but is this explaining anything? So, and the reason why we think lithium is doing that is that this complex formed by beta-arrestin 2, AKT, and PP2A is dissociated in presence of lithium, and that again we don't know why it's dissociated, whether it's cell type specific or what's happening there. So essentially that leaves us with a very simplistic model. Lithium activates AKT, AKT inhibits GSK, lithium inhibits GSK. Solution is found, but that leaves us with a problem. The brain is not a soup. It's not a something in a bowl that we can add product and change activity. It's not freely diffusing. The brain is very complexly organized and is organized at different scales, and the matryoshka story. And when I say that is what is the brain? But the brain is a network of different regions that are connected with each other by complex interconnections. So these are circuits. These circuits are composed of neurons, and each neurons receive innervation and integrate information from several other cells before transmitting this information to several other cells. And inside each neuron, the activity of the neuron is regulated by a complex interaction of cell signaling molecules, which are going to define neuronal activity, number of synapses a neuron will do, whether a neuron will kill or keep a certain synapse, how excitable a neuron will be, and so on. And these cell signaling network are composed of proteins that are encoded by genes, which create a genetic landscape where a little bit of that more, a little less of that, a little more of this will make that this network may have different ability or outcome when stimulated with a certain stimulus, which will then change the activity of the neuron, which will then have repercussion at the level of the whole brain. So what we're trying to understand is how our signaling pathway, our lithium sensitive signaling pathway, is regulating brain activity, and we're trying to look at it at different scales in order to be able to draw certain conclusion about what it does for real. So the questions we have is, but how is this pathway affecting brain function? I mean, where in the brain is it involved? What's the impact on neuronal function? What are the substrates? I will show you one substrate in the next section. How are the drugs affecting this pathway? Is even the regulation of this pathway intersecting with the risk genes that are causing susceptibility to schizophrenia? And the $1 million question, or $1 billion, or billion Bitcoin question, whatever you want, is can we drug this? And so to address this, what we've been doing over the last few years is to take advantage of the CRISPR-Cas9 technology. So essentially what Cas9 is Cas9 is a programmable endonuclease, a molecule that would cleave, in this case, DNA, that is targeted by a specific type of RNA, which is called a small guide RNA, which will correspond to a sequence in the genome. And when this sequence is targeted by Cas9, there is a double strand click cut, so there is a genome damage that is caused by that Cas9, which could be repaired by an insertion if there is a template. That doesn't work very well in non-dividing cells. So what it does mostly in non-dividing cells is that the DNA will be repaired haphazardly by the cell, and that will lead to the formation of indels. And these indels will lead to the formation of premature stop codon, frameshift mutation, and loss of gene function. And this is the method we're using. We're using Cas9 as a blunt tool to cause loss of gene function in specific neural circuit or specific cell types in order to see what it does. So example one of this type of approach, it's a viral vector mediated somatic approach. So essentially this is a technology that is now about 8 years old, which consists of putting Cas9 in one virus and the guide in another using AAV and injecting them in the brain and see what it does. So we were interested in the prefrontal cortex because there's a lot of interest in the striatum, but we published some years ago a map of the D2 expressing cells in the prefrontal cortex. And there's a lot of interesting interaction and circuits and enrichment in region that are involved mostly in limbic function, essentially response to emotional reaction and anxiety-like reaction and so on in the cortex. And these regions of the cortex are driving other brain regions that are involved in regulating fear or emotional response or even associative function in the context of stress. So we are very interested into that. So we moved to use our viral system into this brain area, and we decided to use as a target two things. So GSK3 beta, but we also identified a GSK3 beta substrate, FXR1, which is an RNA binding protein related to the protein that is causing the Fragile X mental retardation disorder. FXR1 is not causing the Fragile X mental retardation disorder, but it's associated by whole genome association study. SNPs in the FXR1 regions are associated with schizophrenia. So it's possible that FXR1 is one of the genes that is affecting susceptibility to develop schizophrenia. And in this study, for example, in human, what we've shown is that if people are I carrier for allele leading to higher expression of FXR1, the control subject display higher emotional stability when tested in a test. However, if they also express high level of GSK3, oops, the higher level of emotional stability are gone, and GSK3 is a negative regulator of FXR1. So we use the CRISPR-Cas9 system to look at the interaction of FXR1 and GSK3 in the prefrontal cortex and try to see what are the consequences. So for that, we use a dual AAV system. The first virus encode SP-Cas9, the good old first generation Cas9, and the second virus is expressing GFP and a small guide RNA, in this case that will target GSK3 beta. And here you can see that if we use the GFP alone, so this is GFP alone vector that is also going to the nucleus, and you can see that the cell is affected by GFP, and even if Cas9 is there, and does nothing because there's nothing in the guide. However, if we put the guide in there, you see that here the GSK3 is gone from the cell that is positive for the vector expressing the small guide RNA. So we are knocking out gene expression in the tissue. And to look at FXR1, we use another AAV system to simply overexpress FXR1 in the system using viral-driven overexpression. So mice were injected. After 21 days, they were tested, either for immunofluorescence to do that type of things, or behavioral testing, or electrophysiology. And what we saw is that if we overexpress FXR1, mice are becoming less anxious in the open field test. They're also becoming less anxious in a dark light emergent test, and they're also becoming less anxious as defined in the elevated plus maze test. So the base of these three tests is mice will avoid being in the middle of large open space or lit space. So they will avoid the center of the field, they will avoid the light compartment, and they will avoid the open arms. And if the mice are less anxious or given anxiolytics, they are going to go in these regions. And if we overexpress FXR1, we have that. And if we knock out GSK3, we have that. So phenotypically, our two genes are doing the same thing. Either we overexpress the negatively regulated substrate, or we kill the expression of the kinase. Is this compatible with electrophysiology? So we went and patched these same cells. And what we see is that actually overexpression of FXR1 or knockout of GSK3 both have the same effect. They reduce the excitability of excitatory neurotransmission. They do nothing to inhibitory neurotransmission. And they do nothing to the generation of action potential when an action potential is artificially generated. So essentially our cells are less excitable, but they are going to be functioning normally if they get excited. And what does it mean at the level of gene expression? But we ran different Western blot analysis in this case, and what we've shown is that, for example, genes like VGLUT are downregulated, so that's the vesicular transporter for glutamate, are downregulated in the GSK3 knockout, and the FXR1 overexpressor genes, phosphorylation of glutamate receptor, subunit of AMPA glutamate receptor is reduced, which is generally associated with less receptor at the surface. So essentially, we have a dampening of glutamate neurotransmission when we are either knocking out GSK3 or overexpressing FXR1. And we know this is strictly dictated, the behavioral outcome is strictly dictated by neurons of the prefrontal cortex regions involved in the regulation of limbic response. So that's fine. Can we go further? So we decided to see, okay, are these neurons in the prefrontal cortex, are they the neurons that express D2, or are they something completely different? So now the objective was to go and knock out GSK3 only in D2 expressing neurons of the prefrontal cortex. And essentially the reason for that is that D2 is signaling to beta-arrestin, to AKT, to eventually activate GSK. So let's knock out GSK3 in the D2 neurons and see what it does. And you can see here that the expression of D2 in the region we're targeting. So for that, we use a Cas9 flux mouse in which essentially the expression of Cas9 was dependent upon the co-expression with a Cree. And in this case, we use a D2 Cree, which is the system that is here. So what we have now is that only the D2 expressing cells are going to express Cas9. And if we inject our virus in the prefrontal cortex, now Cas9 is going to see the guide. The guide is going to be all over the place, but only the cells that have D2 expressed will have Cas9 and the guide, and then we will have a knockout of GSK3 only in the cells that are expressing D2, which is more or less what we see here. And in this case, we were not able to detect GSK3 protein in cells that were expressing D2 and the guide. So we move from there. This is a further validation of the system. The cell here is expressing the guide. This cell here is not expressing the guide. We have, these cells here are expressing the guide. Essentially, we have the knockout of GSK3 only in some cells, and they do have to express and the guide and the Cas9 in order to lose GSK3. Can we get any biological information out of such a precise intervention? Well, the answer is yes. So essentially removing GSK3 in D2 of the cells of the medial prefrontal cortex reduced anxiety as defined in the open field, in the dark light test, which interestingly is the most lithium sensitive of the test we're using. Not in the open field, not in the elevated plus maze. However, when we made a emotionality score taking into consideration the result in all tests, then the statistical significance appeared, and showing that removing GSK3 from D2 neurons is actually reducing as a slight anxiolytic effect in these mice. What's happening next? So we look at other effects. We saw nothing in cognitive function for exploratory behavior of a novel object. However, the mice have some deficit in cognition and in the dealing with remembering a new object, identifying what is a new object versus an object they saw like a few minutes ago. And they also display some social interaction differences. In this case, they seem to be more avoidant of new partners. So complicated task, we don't know exactly what it means, but the objective mostly of this story was to see how far we can push the tools. So we also used another reporter in this system, which is that the same Cree cells expressing D2 also express a tag ribosome, which can be immunoprecipitated. And now what we can see with this approach is strictly the impact on gene expression that was caused by our knockout of GSK3 in these cells. And we can have the perfect signature of what is removing GSK3 from D2 expressing cell of the median prefrontal cortex does to gene expression. And essentially, we were able to run a RNA-seq on that so that genes were changing. And these genes were involved in synapse assembly, neuronal projection, dendritic spine formation, um, and essentially different functions of the sort. So they should affect neuronal plasticity. We're getting closer. We published another paper after looking at GSK3 and FXR1 in the context of sleep and stress, and also showed that FXR1 overexpression or GSK3 knockout were affecting gene change involved by sleep deprivation. So the tool is working. Can we push the method further and now really look at signaling networks or inside neuronal circuit? And to do that, we started in something a little bit upstream, which is the D2 dopamine receptor itself. So again, our prefrontal cortex D2. These cells also express D1, some of them do. And what we did is we designed a Cas9 approach to go knock out D2 in the prefrontal cortex, either using the Cas9-3 or different viruses, and explore the role of D2 in different cell subtypes, or cell subtypes going to different orientation. So when we knocked out D2 in the prefrontal cortex limbic area, what we saw is a reduction of exploratory locomotion, which is often used as a model for mania or emotionally triggered behavior or emotionally triggered behavioral activation. So essentially, that's going more or less in the same area that the knockout of GSK3, but it's more potent. And what we see is that the mice are exploring less, they're more chill. The effect is also found in female mice, so that's reassuring that it's not a sex-specific effect. Then we looked at the object recognition, and interestingly, the effect we saw with GSK3 knockout was gone in the D2 knockout. So GSK3 does something in D2 expressing cell at the level of cognition, but maybe it's not related to D2 signaling. And the major impact that we saw is a reduction of exploration of the objects by the knockout mice. So again, something indicating that the mice are less reactive to environmental cues, less potentially reactive to a lot of things. We used a model of mania that is current, that is the use of amphetamine. And when we gave amphetamine to normal mice, you can see that there is a nice behavioral activation, while these PFC control D2 knockout mice were absolutely amphetamine amorph. Similar phenotypes are found when people remove D2 in the striatum, but it's a bit surprising that removing D2 in the prefrontal cortex completely destroy locomotor responsiveness to amphetamine. We tried to explore whether there was a contribution of one receptor over the other. So we gave D2 agonists. D2 agonists are causing a drop of activity followed by a rebound of activity. So this drop of activity is due to the reduction of dopamine tones associated with activation of photoreceptors, and the postsynaptic bounce is caused by an activation of receptors on postsynaptic cells, which leads to a minor locomotor activation, which is gone if we remove D2 from the cortex. Same thing with D1 agonists. D1 agonists are much less potent if we remove D2 from the cortex. So what's going on? Which cells are involved? So we try to combine viruses, and in this case we use an AAV CAMK2 CRE to go and activate Cas9 in our flux mice only in cells that are excitatory. And when we do that in excitatory neuron, we found again a reduction of exploratory locomotion. We then ask which one of these neurons, these neurons are excitatory and they project. So we studied the projections of these neurons and found that they project to two major areas, the striatum and the VTA. So they project to the neuron that receive dopamine innervation from the substantia nigra VTA, and to neurons that are producing the dopamine in the VTA. So we did models in which now we injected the guide RNA inside the cortex and the CRE in a retrograde system meaning that now we will remove D2 only in cells that project to the striatum or only in cells that project to the VTA. And when we do that, we see that removing D2 in cells that project to the VTA is not affecting exploratory locomotion, but that removing D2 in cells that are projecting to the striatum is having exactly the same effect than removing D2 in the excitatory neuron of the cortex. So we have here a system in which tell us, okay, probably a part of our phenotype of removing D2 in the cortex is caused by excitatory neuron. Not only it's caused by excitatory neuron, but it's specifically caused by excitatory neuron that do project to the striatum. We test these mice with amphetamine. Interestingly, removing D2 from excitatory neurons had a dampening effect on amphetamine. It was not as potent as removing D2 on all neurons. That could be a limitation of the expression system, or that could be that interneurons also playing a role. That's going to have to be explored. Now, removing D2 in neuron that project to the striatum had exactly the same effect. And interestingly, removing D2 in the cortical neuron that project to the VTA increased locomotor response to amphetamine. So we're starting to sort out region projection specific effect on a phenotype of a gene that is expressed in all cells. So if we give a drug targeting D2, we'll both tell the network to increase its locomotor response to amphetamine and to reduce its locomotor response to amphetamine. What this with the CAMK2 tells us is that the majority, the dominant effect that is masking this effect here is the effect of inhibiting locomotor response. So we also played with D1 in these cells, and this is to show you that removing D1 in the D2 neuron is actually anxiolytic. And so again, interesting thing that will be pursued in the future. So in conclusion, we can do somatic knockouts of CRISPR-Cas9 and start to look at cell signaling pathways inside specific neuronal circuits. And we can do that to interrogate the role of genes that are potentially genetically affected to mental illnesses or to developing the possibility of developing responses to drug. This method is compatible with great spatial and temporal precision. It allows to do behavioral research, electrophysiology, biochemistry, morphology, and gene expression studies. So I would like to thank the students who participated into this, mostly Clementine Quintana, who will be graduating soon, Jivan, who left us for Novartis a year ago, Alessia and Sasha. Alessia is now in Paris, and Sasha is also completing her PhD at the moment. So I would like to thank you all. And is there still anybody around, or you all ran away?
Sure. We couldn't stop listening of this fascinating stories. Thanks a lot for your lecture.
Martinsky is great. I really like it. The last part is brand new for me. It's very interesting. Yeah.
Okay.
So please, who has any questions? Yeah, okay. Yeah, I turned on the microphone.
Рамиля, давай.
Sorry.
No.
I can't do that.
Maybe I can do that.
Try. Sorry, it's the last lecture and I pressing and I can't turn on the microphone. Oh yeah, now it's on.
Thank you. Thank you very much for your interesting topic. I was wondering how long does it take to make CRISPR-Cas viable somatic knockout from the injection to experiment?
3 weeks.
Only 3 weeks?
Only 3 weeks. That's the limiting factor is the ability of the experimenter to perform surgeries. If somebody does 50 mice, 100 mice a week, they're gonna have result fast. If they do trickle the mice, for electrophysiology it's better, for gene expression it's okay, for behavior it's impossible. And so the real limiting factors, it's hard work on injecting mice. The other advantage is that we keep one mouse line. We don't keep like a knockout of this, a knockout of that. Oh, this knockout is not reproducing. This other is. So everybody in the mouse in the lab now is working on the same colony.
So you're just changing guides, yeah?
We're just changing guides or Cree. So the D2 Cree is used for some experiment, but the Cas9 flux plus ribotag can be activated. Next step is to combine with calcium imaging, because what is missing in our thing is the impact of our knockout on the neuronal activity. I mean, we can do it in slice, but we doing it with calcium imaging in the living animal would be the natural next step. That's the thing that the lab is not doing yet, but some people have started doing that.
Martinsky, how reproducible it is? I mean, when you're injecting it, does you sometimes probably it depends on hands, probably, yeah?
It depends on the person or the robot doing the surgery. We never had a robot. In our case, the more specific things were done with an injection system that is using electrophysiology pipettes. So you insert a very teeny pipette in the brain, not an Hamilton syringe. It's an electronic control release, slow release, and students that have problem have been pulling out the pipette too fast. Then you have overflow of virus, leaky pipettes, and then it's a mess. So it really depends. It's like macrodialysis. I mean, if somebody's sloppy putting the probe, you'll have whatever.
Oh yeah. So okay, I understand.
A lot more of that. I mean, all the optical sensors that are there for different GPCR activation, not just calcium imaging, is the same story. You want to be targeting brain area you're interested in.
Vasily, you wanted to say something?
Yeah, I have some question if you don't mind. First of all, thank you so much for a very interesting presentation. And you mentioned about antipsychotic action of lithium. It's a very huge topic, but I have some brief question. Are there any difference in the pharmacological action of lithium isotopes 6 and 7? There are at least 2 stable lithium isotopes. And if so, maybe you know something about it and we can use it somehow, maybe.
It's the first time I hear the question. I have no idea.
Oh, sorry. But anyway, thank you so much.
Any more questions? Anyone?
Okay.
No? Everyone a bit exhausted with the
Yeah,
information.
2 days of a lot of information came out. You know, I guess people kind of tired.
Well, I get it. Yeah, but really beautiful, all of the lectures. And
yeah, I would like to thank everybody, all speakers, for beautiful collection of lectures, and students who participated actively and asked a lot of questions. And I would like also to thank organizers, Zhenya first, yeah, and Kirill, Olesya, Natalia, who else?
Zoya.
Zoya. Yeah, you guys did a great job.
Thanks a lot. Thanks to all the listeners. I actually have one thing I want to do. I would like to ask all the students to turn on the cameras. Well, if you can do that, so that we could make a photo, at least an online photo. Не могли бы вы, коллеги, включить камеры, чтобы мы могли запечатлеть да, спасибо большое, чтобы можно было сделать общую фотографию наших слушателей. Спасибо вам всем, что вы принимали участие в нашей школе. Правда, это очень важно. И очень важно, конечно же, не только собрать лекторов, но и то, что были слушатели. Так, сейчас я справлюсь. Так, раз. И сейчас еще одно сделаем. Okay, yeah, great. So thanks a lot for everybody
Спасибо.
joining us and to listening, and surely for our lecturers to giving these great talks.
Okay.
So yeah, thanks, bye, and see you next year, I believe.
Yeah, well, that's great story. We'll continue next year. Hopefully Martinsky will bring you here next year.
Let's hope everybody can travel next year. That's
Yeah, let's hope. Okay. Ciao, ciao.
I need a fix. Okay.
Ладно. Пока всем.
Okay, thank you. Take care, everyone. Keep in touch.